Hello, everyone. I'm really happy to be talking to you today for the US Poultry and Ag funded research, one of our project. So this research, we talk about the Campylobacter vaccine development. Here is the outline. So first, I will give you a very brief introduction to talk about how important the Campylobacter for the foodborne illness. And then we will discuss why our poultry industry need to care about this pathogen. And then I will briefly talk about the previous research for the prevention of this pathogen. And also we will analyze why right now we still don't have our commercial vaccine to control this pathogen. And I will emphasize our research in Mississippi State, so how we fill this gap. And later on, the last one is our follow-up research and our future plan. All right, Campylobacter jejuni is a pathogen, and it causes acute gastroenteritis. The illness name is Campylobacter rhesus. And according to CDC report, this bacteria is the #1 cause of bacterial diarrhea illness in the United States. So each year, there are around 1.5 million cases of infection. This is a high number. So I am a scientist. I like the data. So let's look at the data. So for the CDC report of year 2021, this is a laboratory diagnosed infection. So among these 4 important foodborne pathogens, Campylobacter has the highest number of infections and the highest number of hospitalization and highest number of the infection rate. And talk about the economic impact. So the Campylobacter can cause around $1.4 to $6.9 million loss each year, according to this illness. So why the poultry industry should care about Campylobacter? That's because the poultry is the natural reservoir for this pathogen. And the chicken body temperature is 42 degrees Celsius, so that it provides the perfect growth condition for this pathogen. And also, this pathogen is harm for human, but does not hurt chicken. So this pathogen can asymptomatically colonize the bird in their gut. And also, normally with a very high number, it can reach 6 to 8 log per gram of gut content. So with this high number of germ in the gut, chicken gut, the chicken meat is very easy to be contaminated during the processing and the packaging process. So from our previous research, we found around 45 to 60% chicken meat from the grocery store is contaminated with Campylobacter. And the poultry is not only is the reservoir of this pathogen, poultry is the major source of human infection too. So let's look at the data again. So this is the CDC report for the foodborne outbreaks in 2019. So it's according to different food sources related to beef, pork, chicken, turkey, and other meat. And 4 critical pathogens: Salmonella, E. coli, O157, Listeria, and the Campylobacter. So let's look at the chicken. So look at chicken. The Salmonella has about 20% outbreak come from the chicken meat. And E. coli, O157, and Listeria is around 0, so we don't so care about that. But look at the Campylobacter. More than 6% of human outbreak related to eat the chicken, contaminated chicken meat. And for the turkey, it's about 5%. Totally more than 65% is a pretty high number. So that's why our poultry industry and our university has the responsibility to reduce this pathogen and keep the consumer safe. Of course, we have already applied different intervention strategies in the pre-harvest side and the post-harvest side. So it includes the managemental practices, including biosecurity, litter management, and water treatment. And also we have different feed additives, including prebiotics and probiotics, try to reduce the gut colonization of Campylobacter. Of course, vaccination is a hard part for the research side. And in the post-harvest side, So we have the plant cleaning and the sanitation, and the carcass decontamination with different antimicrobials. But the chicken, live chicken, is the contamination source. So we want to target the Campylobacter jejuni at their source. So we want to focus on the pre-harvest side. So we know the fewer pathogen coming into the plant, the fewer pathogen will be in the plant. The same thing for the meat. The fewer pathogen will be in the meat. So research report, if we can reduce the chicken Zika colonization by 3 logs, so we can reduce the risk of human infection by 90 to 100%. So that is incredible. This is why we want to focus on the vaccination method to reduce the chicken colonization. Okay, let's talk about the vaccine history. Actually, start from 1990s. There's a research group study the live attenuated Campylobacter jejuni vaccine, and around 30 years right now. So different type of vaccine already being developed, including the killed whole cell vaccine, protein subunit vaccine, the recombinant vector vaccine, and the polysaccharide conjugate vaccine, and also nucleic acid vaccine. However, although we have so many research studies, there's no one commercial product provided to the poultry industry to reduce the Campylobacter colonization. So let's analyze what's the reason, why, what's the gap of knowledge. So let's go back and look at the traditional vaccine development approach. So use the traditional method, we start the vaccine development from the live bacteria. So we isolate different components from the live bacteria and purify them and try to analyze their properties, their function, to see if they can cause the immune response from the bird. And if we failed, we go back to work again. So this is a circle procedure. It takes a very long time. And if success, so we go to the next step. We clone this gene. I talk about subunit vaccine. Clone the gene and do the immunogenicity test, and then to develop a vaccine. However, this method has a major issue is these antigens come from one single strain of bacteria or a field. So when we go to the field, so the chicken will face to a very complicated environment. Unfortunately, the Campylobacter jejuni has more than 50 different serotypes. So in the field, normally this vaccine will not work to reduce the colonization of the Campylobacter in the chicken gut. And another concern is there's always new virulent strains emergence in the field. So this vaccine will not work for the new emergence vaccine. That's the gap of knowledge for our analysis. So here we have this gap. How can we fill this gap? So can we develop a universal vaccine can fight against all different strains of the Campylobacter? So traditional method so far is failed. So we need a new approach. So this is the purpose why we share these slides with you to emphasize our new approach. So the approach we used called reverse vaccinology. So the major difference compared with the traditional vaccinology method is this one is start from the whole genome sequence of the pathogen, not start from the live pathogen. And the biggest difference is the antigen selection procedure different. After we got the antigen select, the rest part is the same. So we do the immunology test and to develop the vaccine. But this method can really reduce the antigen selection time. We use 1 or 2 years, we can select many antigens. Okay, talk about the advantage of this technology. So because we know the whole genome sequence of the Campylobacter, technically every single antigen can be selected. So then we use the computer program to analyze these antigens. Then we can predict which one is our better candidate for the future usage. Now let's talk about how we use this technology to make a universal vaccine. The traditional vaccine, we use single strain or a few strain, but for reverse vaccinology, because we have the whole genome sequencing technology very good right now, we can sequence a lot of different strains, and we have very good database, NCBI database. We can all upload our sequence to the database. It's shared, it's free for everyone. So we can use all these data at the very beginning, at the first step, and try to identify the core genome. The core genome means the shared sequence in all different strains. And then start from the core genome, we use different computer prediction tools and to use the in silico method to identify the vaccine candidates. Okay, the goal for this research is to develop a universal Campylobacter vaccine. So I separate this goal into 6 different objectives. And before we apply this US Poultry and Egg grant, we already studied isolate a few Campylobacter jejuni strains. We already isolate 66 strains. And since this project is only 1 year, so we proposed 3 different objectives in this US Poultry and Egg grant here. So it's whole genome sequencing, and the second one is bioinformatic analysis, and third one is antigen selection. So the last 2 actually now include in this presentation. So now let's move on to the research at Mississippi State. The first step, of course, the whole genome sequencing is very important for using this reverse vaccinology, because if we don't have the high quality genome sequencing, we may have the wrong result. Since we don't have many Campylobacter jejuni sequence in the database, and at that time we searched, we have only 18 Campylobacter jejuni sequence from poultry source. So that's why we decided to sequence our own isolate first. So this is the procedure for our whole genome sequencing. So these isolate was isolated from the live bird and from the grocery store, from meat, and we isolate the single colony. Because we have 66 isolate, we're not able to sequence all of them. So we start test their antimicrobial resistance genes and their antimicrobial susceptibility test. Then finally we select 4 strains for the sequencing. And we extract the whole genome DNA and then sequence using the short-read Illumina sequencing platform and long-read nanopore sequencing platform. The goal, we use 2 different platforms because we want a completed whole genome sequence. So they have their advantage and disadvantage. So after sequence, we form the assembling the sequencing, we get the complete whole genome sequencing. This is 1 strain. And this part of research we already published in the microbiology resource announcements. So here is the summary of the genome sequence. So you can see we have 6 different section number here. That's because there's 2 isolate carry plasmid. So that's why we have 6 section number right here. But the chromosome we have 4. Okay. In 2020, we finished this whole genome sequencing work, and then we move on to the next one, bioinformatic analysis in 2021. So for this, this is our key technique in this grant, in this project. So it includes 6 different steps. So the first step, so we talk about the universal vaccine prediction, right? So first, we input all 4 different C. jejuni whole genome sequences, and we use the tool to pull their core genome out. And then eventually we get our result with 1,375 protein sequences that shows up in all 4 different Campylobacter jejuni genome. And then we use different tools to predict their outer membrane predictions. So we use the P-sort-B. This one is for us to predict if these protein are extracellular or on membrane proteins. Because for a better interaction with the host cells, these proteins need to be in the out membrane side or extracellular side. They have a better communication with the host cell. And then we use these TM, HMM tools to predict their transmembrane helix, to predict the protein secondary structures. Because our goal is to produce the subunit vaccine, eventually we will transform this one to E. coli and produce the recombinant protein. If we have too many transmembrane helix, it's hard to purify, hard to express. So we select the transmembrane helix less than 1, 0 or 1. And then we use the SPAN tool to predict adhesion probability. The number is higher than 0.5. And then next step, we use the Vaxigen V2.0 to predict their antigenicity. So the score we set up is the antigen score greater than 0.5. And then we end up with eventually 27 protein sequence here from the step 3. And step 4 is the conservation and homologous analysis. First one, we want to know our candidate is different with even if the poultry genome, because if they have similarity too high, they may cause the autoimmune response. So first, we blast these sequence with Gallus database and make sure they are not similar. And then later on, we want to do a really universal vaccine. So we pull the C. jejuni genome from the NCBI. We find 206 different genomes. And also we want to see how similar they are to the Campylobacter coli, not only jejuni, because coli and jejuni are the two major concerns for Campylobacter infection. So right now they have 34 C. coli genome available in the database. So after compare with these whole database, we end up at 21 protein sequences. Next step is the B cell epitope prediction. So we set up the B cell epitope density greater than 0.01. And then the last alignment, we end up our final product with 6 prospective vaccine candidate. So here is the table for these 6 vaccine candidates. So this is BTUB, is CDTB, FLGE, FLRD, MONP, and the PLDA. There are different proteins in the outside membrane of the Campylobacter jejuni. So when we get these candidates, we want to know if these candidates are started alright or already, or they are new, they are novel antigen, nobody started. So we found these 3, FLGE, FLD, and MONP, already been started. So later on, we target these new antigens as nobody applied them into the vaccine development for Campylobacter jejuni. It is BTUB, CTTB, and PLDA. So with this step, we have 6 conserved antigen identified. Eventually, we select 3 for the next research. So next step is the antigen selection. So we want to know, because the second step is in silico work. This step, we want to use a cell model to verify if these candidates really have some function during the pathogen-host interaction. So in this step, we use a AVEN cell called AVEN HD11 cell to study the pathogen-host interaction analysis. The key point is what we want to know during their interaction, if these 3 candidates gene expression can be modified. If they are highly induced, that means they have some function during the pathogenesis of the Campylobacter jejuni. So we use the adhesion and evasion model. This is the most popular model for the pathogen-host interaction analysis. And for this part, we use so let's briefly introduce the procedure. So we mix the C. jejuni with cultured AVEN cells, and then culture for a few hours. And then we wash the unattached cell. Then the attached cell was counted by the plating. And later on, the invasion cell was counted by the plating too. And then during their interaction, the pathogen should have some response to the interaction. We want to know if the vaccine candidate gene expression can be modified. And also we are curious about the host immune response. So these two things we use real-time qPCR to identify them. First, let's talk about the adhesion and invasion. So the results showed, so in this table, in this figure, ADH means adhesion, INV means invasion, and the y-axis is the log number of the bacteria. So the adhesion number is around 6 log, and the invasion is around 4.3 log. So we transform it into the percentage. About 3% Campylobacter jejuni can adhere to the avian cell, and about 0.1% can invade into the avian cell. So after this one, we try to analyze the pathogen response. So this is the key slides in these objectives. So first, let's look at the result. So in the x-axis, NTC means the negative control. TRT ADH means the treatment adhesion. Why we select adhesion? Because the invasion number is too low. The RNA is not enough to run the qPCR. So the y-axis means the relative fold change. So from this result, you can see all three genes, CDTB, PLDA, and BTOB, are highly induced during the host-pathogen interaction. This means these three genes may have some function for their pathogenesis. They may be a very good candidate for the vaccine development. And this step, actually, we spend a very long time to identify that. That's because in the adhesion here, you can see they are mixed system, include host cell, includes pathogen cell. So when we extract RNA, we cannot separate which one prokaryote, which one eukaryote. We use about 2 months to identify, to quantify the amount of prokaryote RNA, then to do a real-time PCR. So this one, I think it will help for our pathogen-host interaction in the poultry science. If anyone interested with this research, we can talk about it later. Mm-hmm. And then we test about the host immune response. Normally we say, okay, Campylobacter does not affect the host, but we do find the host has a response. So we test the pro-inflammatory cytokine gene IF-GAM and the anti-inflammatory cytokine gene IL-10 and our receptor gene TLR2. So IF-GAM and IL-10 are highly induced, and IL-2 are significantly lower. This we don't have no answer, but they do have interaction. From now on, all the research part done. So I want to report our outcomes. So first of all, we finished all 3 objectives. We have 3 potential vaccine candidates identified. So these 3 candidates are localized in out-membrane protein or the extracellular protein. And they carry the antigenicity capacities. And also during the host-pathogen interaction, they're highly induced. This means they have the potential to be a good candidate for the future Campylobacter vaccine development. And also we sequence 4 C. jejuni genome. And these 4 are already deposited to the NCBI. Now it's free for everyone to download and use them. And also through this project, we verified this novel approach can be used for vaccine antigen selection. It's quick and efficient. It's not only can use for C. jejuni, it can use for any other bacterial, virus, pathogen. Right now, we use this method for even pathogenic E. coli vaccine development. And also for the other outcomes, so the output, we already published 2 journal articles, and there's one submitted to Poultry Science. It's under review. And this project, we have 5 abstracts and 3 oral presentations. And 1 PhD and 1 master were trained by this project. So for taking this opportunity, I want to introduce my graduate student, Sabine Perdue. I didn't see him today. He's the one mainly work on this project, and he presented these 2 different meetings and have 4 research awards right now. It's amazing. And he's going to graduate this 4th semester. If any university, industry interest with this technology, technique, so, and has position open, he will be the perfect one, perfect candidate for you. And let's talk about the future research. So now, with the support of US Poultry and Ag, we already finished these 3 objectives, and we have 2 more left. And this one, actually, we already got funded by our main phase, Mississippi Agriculture and Forestry Experimental Station. So our goal is until 2025, we have 1 or 2 products can be verified in the lab. Of course, to make a really effective vaccine is challenging. It cannot be finished by 1 lab or 1 person. So I'm looking forward to collaborate with the industry or university collaborators in the future. So we work through this one and reach the goal. I would like to acknowledge our funding resource, US Poultry and Ag Association and the Mississippi State, Mississippi Agriculture and Forestry Experimental Station, and our awesome co-PIs, Dr. Pritima Adhikari from Mississippi State and Dr. Aaron Keys, now from NC State. And my students, Sabine Purdue, Tianmin Li, and Dr. Linan Jia. And our awesome collaborators, Dr. Changyu Su, Mark Eric from IGBB. So thank you for your attention, and